Great. Welcome to RBCM at Home. My name is Kim Goff. I'm a learning program developer at the Royal BC Museum. I hope everyone's doing well and finding ways to take care of themselves. And also I want to thank you for watching us and tuning in, making us part of your online diet. I hope you find it's nutritious and some good use of your time, your screen time. I'm coming to you from my home on the territories of the Lekwungen speaking people in Victoria. Like many of you, staff at the museum has been asked to work from home during this pandemic, and in this series, At Home, we explore what that looks like. We'll visit with members of the curatorial and collection staff and discover how they do their work and some of the research that they've been working on. Today, we have two presenters to discuss our topic, which is documenting marine diversity. Dr. Gustav Paulay is a curator at the Florida Museum of Natural History and a researcher at Friday Harbor Laboratories. His interests are in cells and molecular biology and marine invertebrate zoology. Dr. Henry Choong is the curator of invertebrate zoology at the Royal BC Museum. Henry's work on BC's natural history includes the discovery of several new species of hydroids off Vancouver Island. Henry, let's start with you. Documenting marine diversity sounds like an impossibly large task. Where do you even get started with something like so big as this?
Well, it is actually a humongous task, and I think that most people would be amazed to hear that we don't know much more than we know. So there's a lot that we don't know, and the other thing that strikes me is that it's very easy at a casual glance to think that we probably know, we know everything about most of the animals on the shoreline, and because at a casual glance it seems like it doesn't change very much from day to day. But when you really start to dig deep, you find that there are lots of things that you don't know, and every time we go out, we find species that we've never seen before. So where do you start? You start simply by going out and looking. And we also find that some groups are better covered than others. And in the groups that I work with hydrozoans, for example, with hydroids, a lot of it is still wide open in that there are lots of things we don't know about them, and so on. And so, but I'm really happy today to have Gustav on board, because Gustav, I've been very privileged to have gone out with him a few times within the last 3 years or so. And every time we go out, I learn new things. And Gustav has collected all over the world, and he knows many different groups.
So partnering is important, having collaborators, because this is such an enormous task. As you're saying, you just know, you have like a drop in the bucket of what there is to know, but you start pulling together people from all these different areas. And
yes, it is. And that's a good time for me to actually pull up the screen here. And I won't show these slides necessarily in sequence, but oh, here we go. So, yes, so speaking of collaborations here, whenever we go out, I think the other thing that I would like people to know is that these are group efforts. So there's a large number of people involved in these efforts. Again, because the diversity out there is so large, many of us specialize in certain groups, and so it really helps when you have different people on board on these trips who are familiar with different groups, and that way you cover more ground, so to speak. And it also makes it fun, because you get to see the people you know, some people you haven't met before, and everybody brings their expertise on board.
excellent. Was this your first collaboration together, this 2017 Hakai BioBlitz?
This was our first trip together, yes.
Great. And Gustav, what are you doing on these ships? What are you looking for? What is your main area of interest?
Well, I'm really interested in marine diversity as a whole. And I guess if I have a specialty, it is to sort of organize these large scale efforts by bringing people together for it. And as Henry said, they are really major group efforts. You have a lot of people who are out there helping with the general logistics of it, you know, especially if you go to a remote area like up at Calvert Island, where the HECA Institute has an incredible facility. And then there's a lot of students and techs and just, you know, volunteers who help with finding things and sorting things and identifying things at a basic level with us. And it's a really fun task. You can imagine just going out there looking for critters. That's always a fun thing. And then we have, of course, a lot of taxonomic specialists who really know a group or another incredibly well, like Henry with hydroids. And they look for their specialties. And of course, they are able to sort of sort out from the catch when it comes back what's in it. I mean, just imagine bringing in a bucket of seaweed or mud or rocks, and there's hundreds and hundreds of species in every one of these. And the only way you can figure out what's in there is to have people who know those animals to tell them apart. So you can recognize and individually document each one. And there's just huge amount of interesting stuff you come across, including things that we know, including things that we think we know, but we don't really know. And of course, a lot of things that are completely new to us. And it's really a very adrenaline-rich, wonderful time. It's like a couple of weeks long party when we have one of these things going on.
So is a BioBlitz the main way of getting this sort of bulk of information?
I mean, the word BioBlitz has a tortuous history, but basically the way it's employed here, yeah, it's the best way to do it, because if you send individuals out, they usually focus on a single group, and certain groups have become very well known. You can imagine sea stars have been studied a lot, although we're finding out even they are not that well known. So if a sea star person goes out, or if Henry goes out looking for hydroids, we'll get to know the sea stars or the hydroids. But there's so many groups together that the best way to hit them is in large groups where you capitalize on the person power of many individuals picking things and the expertise of specialists who know different groups pretty well.
Excellent. So you were saying that this is Calvert Island. We have some folks here, not from BC. Do you want to explain where Calvert Island is, Henry?
Yes, Calvert Island is on the central coast of BC, and Calvert was originally in recent times a fishing lodge on Calvert, and that was turned into a research center by the HECA Institute, by the Tula Foundation, run by Eric Peterson and his wife Christina, and so they're interested in what's going on with biodiversity and what's going on with the larger picture in terms of what is happening to the Central Coast, what's happening on Calvert, and how does that fit into the larger picture, and so on. And so one of the things that happened was that in 2017, the HECA Institute and Gustav and the Smithsonian people put together the BioBlitz, where they brought us in all together, a bunch of different taxonomic people to look at biodiversity in this very intensive 3-week period. And so I came on board to look at the hydro zones. And so the interesting thing about Calvert is just have a couple of pictures here of what some of the beaches are like. Is that you have both the outer coast and the inner coast, and so it's a variety of different environments in one space. And within that 3 weeks, for example, you see here, these are some of the red dots are some are the location sample, some of the location sample. So we sampled about 255 places and collected lots of invertebrates.
Was everyone focused on invertebrates on this trip?
Yes, on this particular trip, yes.
Fantastic. So what's a typical day like on one of these BioBlitz? How does it start?
Well, it starts differently for different people. For Gustav, I think the 3 weeks is one long day. So there are different people. And then the beauty of these trips, I think, with such a large team is that you have different people doing different things. You have offshore teams that are away collecting really early in the morning, or depending on the tides and so on. And then for some of us, the day starts when we get up and have breakfast and head to the lab and start to work on material. And also material will start coming in, and you try to get to that as fast as possible, because one of the things that we are doing on these trips, and this looks like, I know it looks a little bit chaotic, but it really is not. Because the way that it's laid out here, when we go in here, so Gustav has a plan for the place. It's all laid out. All the different stations are doing different things. You have, for example, the sorting, the photography, and then on the bottom right there you see that's one of the people working on polychaetes, on worms, so you have the sorting and identification. You have the photography, and you have sampling for tissue and so on. And so that all goes on. And so that goes on through the course of the day. And we try to photograph the animals when they're still alive, because they look very different once they're preserved. And then the localities are documented. And so it's in a way, it's like a processing, it's like an assembly line almost. So you try to do that very intensively. And so that goes on through the day with a break for dinner. And in the evening, we usually have a meeting to talk about what happened during the day and to summarize things and so on. And then it's back to work. So these are usually pretty long days, and they can go on until pretty late at night. And then it starts again the next day.
And you said this trip was about 3 weeks. Gustav, it sounds like your role was a bit like a project manager. How would you describe it?
Yeah, I think that's what it is. I mean, this is kind of what I do. And, as we are realizing how little we know about marine life, these big surveys have become very popular, and I've been doing them in many places. And this Calvert Island one was one of them, a really special one, given where it's at and the amazing environment and biodiversity there. You basically, invite people who bring either expertise in field techniques or in taxonomy, or are really enthusiastic, and you want to expose them, to these methods. And then, as Henry said, you started the day and a bunch of teams go out, and some people are divers and they'll go scuba diving to get animals. Others use remote methods from boats. If it's a good low tide, a lot of people will be up at the crack of dawn or before, because that's when the tides are and they're heading out. And on Calvert Island, there's no vehicles, there's no real roads for vehicles. So you're hiking for a mile or 3 to get to your site. So that's quite a task. And then others who are not so field oriented, but have really deep taxonomic expertise, are just working away back at the labs while this is happening. And the field teams bring back stuff, and is just a nonstop action all day long. And one of the things that a couple of us do is sort of gauge what we got, and who to send out and where to send them out, and what do we need to go after in terms of environment or biota. And based on that, our goal is to cover as much diversity as we can, given the people that are there and the power that they bring to the project.
So I imagine your plan might have to change on the fly. In one of those evening meetings, you see, all of your samples might be representative of one thing and you're not getting what you were hoping to get, so you have to be thinking on your feet? Or is it because there's not a lot of resources, are you kind of stuck?
Well, the resources are amazing with the Hakai Institute. So we weren't challenged by that. Sometimes you are limited by the weather in terms of what you can do, and also by other projects that are running in terms of availability of certain things like boats for diving. But you know, you're changing your plan hourly, not just daily. And you know, you see that there's, you know, you're overwhelmed with material, then okay, stop going in the field, or you don't have enough. It's like, you know, go out again and get them, and yeah, it's fun. It's a lot of fun. But you know, the first time you do one of these, since you're trying to do every species, you can go anywhere and you're inundated with diversity. And you know, we do every species because we're also finding that a lot of things that we think are one thing are turning out to be multiple species once you start applying modern techniques like DNA sequencing to them. So getting the same thing from Calvert Island and Friday Harbor is important because sometimes they turn out not to be the same, even though we thought they were. So we kind of redid all the biodiversity there from scratch just to have that baseline and the comparative basis for understanding that biota.
So a lot of that research, that DNA sequencing and other things you're doing, that's happening back at other institutions. So when you have a big group of people on a BioBlitz, where does that material go afterwards?
Well, the general process is that it all ends up in a collection. So in most of the surveys that I'm involved in, they end up in the Florida Museum, mostly because we have the capacity to process them and to make all the information available online immediately. And I can share a screen with you on that later if you want. The tissues themselves, though, that the DNA sequences come from, end up in one of the high throughput labs. And in the Hakai example, they were pre-processed by the Hakai personnel, and then they were sent to the University of Guelph, to their very, very famous barcoding center for sequencing. In some of the other BioBlitzes I've been involved in, they'd be sent to another facility. For example, the Smithsonian has a large robotic sequencing facility that does that. And then we also take photographs of everything, and the photographs get digitized, and then of course everybody can have that, and we put them online as well. And then the DNA sequence data, when it comes back, ends up in the Barcode of Life database, which is a public open database also run by the University of Guelph. And that goes online. So all of the information, the records of the specimens are available from collection databases, and online the photos are and the sequences are. So all of that becomes public very quickly.
Amazing. There's a question that came through via Facebook. Amber Diffie is wondering, are all your samples from the shores or do you dive?
Oh, a lot of us dive. I dive. And so we did as much diving as we could. And not in 2017, but in 2019, neither Henry, you weren't there in 2019, were you?
On Hakai?
Yeah.
No.
No, neither of us were there. We were actually, both of us were in Oregon on a different BioBlitz.
Yeah, that's right.
But in 2019, the Hakai Institute folks did a deeper survey with some ROVs and deep dredging. So they got some deep water material from that. And we are right now in the process of analyzing those samples as well. But we weren't ourselves participating in the fieldwork on that, just in the workup of the samples afterwards.
So why is documenting biodiversity so important?
Well, let's put it this way. If you were in Rome in about 400 and you went into the great library of that Romans had, I'm sure they had some amazing resources. All the knowledge of the classical world was there, right? And then all of a sudden the barbarians came in and burnt Rome down. And we had 1,000 years of dark ages. That's what we're doing to the planet today. And we're gonna lose a lot of biodiversity, even if we manage to mitigate as much as we are trying all the impacts that humans are having. So understanding what's in that library is really important. That's, 4 billion years of evolution, millions and millions of species that have learned how to make a living. And we're losing all that knowledge that's present in their biology, in their genomes, in everything. And right now is the last chance to do that, to capture that knowledge for many things, because they're not gonna be here tomorrow. So that's what drives me. And as we are learning, as we do this, we learn how little we know. I mean, the slide that Henry has up is a sea star, the blood star, very common. We thought we had 6 species of them. We applied genetics to it, and there are 23 now, okay? So nothing is as common and well-known as a sea star. And yet we know very little about them. So, and if they disappear for global warming, acidification, what have you, we'll never know what stories they have to tell and what incredible knowledge they could impart to humanity that could change our lives.
See, it does sound like a very urgent project. So how many BioBlitzes are you able to participate in a year, for example?
It varies. Typically, I try to do 2 or 3 a year.
Amazing. And Henry, what about you?
I think 2 or 3 is a pretty hefty number. So usually for me, it's about 2 a year for the bigger ones. At least it has been for the past few years with Gustav. And then in between, I have quite a few smaller trips, shorter trips that I go on as well during the year. I should also say that, in terms of where these weighted collections go, that the Royal BC Museum has a subset of the hydrozoans that are collected from on these trips as well. So as Gustav was saying, that the work doesn't end when the BioBlitz is finished. So the work is always going on, and in terms of identification, because a lot of these, like with the sea stars, that a lot of these species look very much alike morphologically, and in the fields you can't always tell precisely what they are. So we come back and revise, in some cases revise our field identifications, our findings, and so on. And so I do that with the hydrozoans. So I have a subset of the hydrozoans at the Royal BC Museum. And again, the other important thing is that we are all sharing the data and we all contribute towards the final picture.
So important. So where do you want to go next, and what are you hoping to find?
Gustav?
Wherever the next project is. So right now I have a pretty large project that got funded by the US NSF Fund, US National Science Foundation in Oman, in the Middle East, which is a very unusual area in terms of its biotic diversity. So we have a lot of work going on there. I'm in Florida, so we do a fair bit of work there. But another focus is really the Northeast Pacific, because I spend a large chunk of the year at the Friday Harbor Labs where my wife works. And so we've been doing a lot of BioBlitzes on the West Coast. We did one in Oregon last year, another one in the inner Puget Sound area with the Washington Department of Ecology, and another in LA. So we are hoping to continue that and basically cover the coast from Southern California through Alaska and get a much better understanding of the West Coast marine biota through repeated surveys. Typical BioBlitz like this gets you a good 1,000 species, but the total fauna is on the order of 15,000 species. So it'll take a number of efforts to start accumulating that knowledge at this new modern, DNA level and photography level and all that. And there has been so little collecting done in recent years that it's really important to go at it again, because many of these species are barely, if at all, present in collections. And so there's no material to study to speak of.
Henry, are there any slides that you want to show us next? If not, I will ask you to stop sharing your screen, or you can take us to a slide.
Okay. Gustav, did you want to say a little bit about the phoronids before I...
Sure, show the phoronids picture. Right. Why not? So one of the fun things we found in Calvert was a field of these worms. There's a nudibranch that's gumping down on them in the middle. But those fuzzy things are big tentacles of a worm called the phoronid. And phoronids are a phylum. So a phylum is like mollusks and arthropods and chordates, which include, among other things, sea squirts and vertebrates. So this is an entire phylum. And there are 13 species of phoronids known in the world. And so they were just everywhere in Hecai. So we started sequencing phoronids, and so far we've got just a little data back, and one species we thought was one turned out to be 3 with DNA. So then I started looking at what else has been sequenced in the world and our other data, and we now have over 30 species of phoronids based on DNA, and globally the known diversity is 13 species. So here's an example of how little we know. This is one of the most important marine animals in some ecosystems at Calvert Island, and Globally, we think we have 13 species, and out of 109 specimens people have got DNA out of, there were more than 30 species represented in there.
Wow.
Okay, I can stop the screen now. Yeah, there we go.
There is a question here in our Q&A window, and the question is, what do you do with the non-invertebrate species that you find? Do you share them with cross-disciplinary colleagues?
You take it, I take it?
Sure, go ahead.
So it really depends on the project. So when we are in a place where there's non-invertebrate workers along with us, then we share specimens between teams. The problem really, I mean, invertebrates are approximately 75% of the macroscopic diversity in the ocean. So it's really most things. And fishes, which are the only other animals, are generally pretty well known, at least in this part of the world. So they don't, and they've been studied at pretty deeply in enough places that we don't usually pay attention to them too much because they're well known. Algae are a different story. There's a lot there. And in Calvert, there has been a lot of phycological research going on by other teams independent of this BioBlitz. So they are getting covered really thoroughly by the phycologists. Really tiny things, protists, are a whole other ballgame. And again, that requires really specialized expertise and techniques, microscopy, and so forth. And the University of British Columbia has some incredible people who work on those. And they've also been up at Calvert, so there's actually a developing database on the protists of that area.
Henry, were you going to add something?
Yeah, well, in terms of, you know, I forgot what I was going to say. No, I think it seems like I think one might get the impression that you go out there and, you know, you're scooping up everything. In a way, we are, but it's really, it sounds contradictory, but it is quite targeted. So in other words, when we go out looking for invertebrates, we generally do not collect. You don't, for example, collect a fish for no reason. So I just wanted to make that clear that you don't do that. The other thing that I wanted to say was that, you know, in talking about invertebrates, that a lot of invertebrates will grow on top of or live on top of one another. So, you know, when you're getting a sample, you know, one sample of one thing could potentially bring you many different whole community of organisms to study. So there's lots of things out there, and I see that a lot with hydroids, because hydroids will grow on other hydroids, and the protists on the hydroids, and we don't know a lot about some of these associations, you know, and how specific they are, and so on. So there are lots of things that we're still learning from those.
I have one final question, and it's hard to answer because we don't know what's happening these days or what it might be like next summer or this summer. But we have a lot of folks watching who are perhaps not professional scientists. Are there opportunities for the public to take part in these BioBlitzes? Is there a role for a person with general knowledge or very little scientific knowledge at one of these events?
The answer is yes. There are many different kinds of BioBlitzes. Some of the trips that we go on are little more intense and mostly a very sustained effort in terms of specialists who are collecting. There are other types of BioBlitzes who, for example, in 2016, we did a lot of BioBlitzes with Parks Canada. 2016, 2017, we did a lot of BioBlitzes with Parks Canada, and those involved a lot of public effort in terms of people being able to participate or simply being there to observe what's going on. So the answer is yes, and it depends on what kind of BioBlitz it is.
But the term BioBlitz originated from a public angle. It was coined for these efforts that the National Park Service and National Geographic ran for 24 hours in an area where they wanted anybody and everybody to come together and record as many species as they could in a 24-hour period. So, and they still continue those kinds of BioBlitzes. And in any of these big ones, interested people are always welcome. And we often have some pretty good participation from amateur naturalists who are quite knowledgeable.
And where could someone who is interested, where might they look to find out information about upcoming?
Well, I think you would want to look for your neighboring field stations and research institutes and see if they're sponsoring one. The other opportunity is to have somebody like the Royal BC Museum organize one just for the public. And then, you know, a couple of us would be happy to come over and roll around in the mud for a day with you.
Well, let's talk about that, Henry.
Sounds good.
Thank you so much, Gustav, for joining us, and Henry for joining us and suggesting this topic and introducing us all to Gustav and some of your colleagues and the work that you all do in documenting this biodiversity, which is, as you've really well articulated, an important task and something we need to be doing. So thank you for doing it. To mention, there is a learning portal pathway that talks about some of this. It is called the Great Bear Rainforest, and it includes information from a BioBlitz done there. So if you want to visit the learning portal, go to the Royal BC Museum webpage, and you can follow it from there, and you can see more great pictures and learn more information about that. RBCM at Home takes place every Tuesday and Thursday at noon. And you can join us next Tuesday when we chat with Curator of Entomology, Dr. Joel Gibson. And he'll talk about the diversity of insects and spiders that you can find in an urban yard. So we're going from the massive ocean and a BioBlitz just to your own backyard. So maybe it's like a mini BioBlitz you can do in your backyard. Joel will talk about how you can do that and what you might expect to find there. If you have young people in your homes, let them know about RBCM at Home Kids on Wednesday at 11. These sessions feature a museum educator and a museum staff member who share activities that you can do at home. And also on Wednesdays, we have RBCM Home Outside. Next Wednesday, I believe, Henry's collection manager, Heidi Gartner, is going to take us to Willows Beach. And so she'll be talking about what you can see in the intertidal zone around here in Victoria. Thank you all for joining us today. Please take care of yourselves and take care of one another. Thank you.
Thank you all. Thank you, Gustav. Good to see you.
Thanks, y'all.